Good afternoon, everyone. I think most of you guys are in Korea. For me, it's morning. My name is Zinah, and I am a postdoctoral researcher from 성균관 University. Before I start, I'd like to thank Brick for giving me the opportunity to talk about my research latest publication that was published in PNAS. The title is Design of Light and Calcium Switchable Organic Peptide Hybrid. So to start with that, the contents will be, I'd like to first start with the platform of what is protein design, and then what is metalloprotein, and the metal I used was calcium indicator development, and then I'll talk about the resultant discussion throughout the summary and then acknowledgement part. So what is the principle of protein design? Most of us know that proteins consist of sequences of amino acid residues, and those amino acids, they will fold into a certain kind of a structure to give a protein, and that structure will be given a function. Yet this was the old way of designing proteins. It's quite difficult because we cannot predict the structure from the sequence at the get-go, from the beginning. So researchers, what they started to do is start from the backward, basically decide what kind of a function we need that protein or peptide to design with, and then pick up the structure that fit that function, and then we can choose the sequences that could make that structure fold. So this is the basic of protein design that people follow nowadays. Now, what is the protein design from the function, as I said previously? What can we do with it? It can go from three main categories. First one, we can just design functional de novo proteins that can bind with metals. Basically, we can pick up a metal and then we can accumulate a certain kind of a structure that can bind with it. And the second thing, we can design protein binder, including ligand binding, protein-protein binding, or protein self-assembly. And the other last one, which is the one that what we do the protein design for, is for a biomedical application. The recent one is COVID, as you all know. So these are the three main functions that design that can go from the function for protein. Now, with that being said, the protein that I'm gonna use, I'll be calling the terminology metalloprotein. So if you don't know, I would just like briefly introduce you to it. It's simply any kind of a protein that contains a metal cofactor within its own structure. Now that protein can give that metal can give this protein a kind of a function that could be helpful for like biological processes, for example, enzymatic catalysts and neuronal activity, etc. Then what can this metal do to this protein? Basically, as I mentioned in my first slide, there are three things we need: is structure and function with the sequence. Basically, if we have any kind of a protein secondary structure that is kind of not loose or not stable, we can introduce a certain kind of a metal to that structure, and that metal may give it a new kind of a folding. Basically, that fold will give it a new function. So basically, the metal will assist this protein into a new activity, or it can be used as a catalyst for that matter. Now, the metal I used for my work was calcium from the title. So every project does have a motive for its work. And for me, the reason why I picked up calcium to design for it was for neuronal activity purpose. Basically, calcium is quite important for neuronal activity. It can transform the signal from one neuronal cell to another throughout the synapse area. Yet, if there is any kind of dissociation within the calcium in the brain, that will give us a new kind of diseases like Alzheimer, Parkinson, and a lot of other area. Now, developing a new tool or a device that can detect the calcium activity within the brain is quite important. For me, I wanted to take that as a start for designing calcium indicator for the brain activity. So next to that, to start something new, we need to go from what is the most common thing they use for calcium indicators. There is a protein called GCAM. It's a synthetic fusion between this is a GFP, a fluorescent protein, and with the comadoline, which is calcium binding gene. As you can see from here, before calcium was introduced, the comadoline didn't have a certain kind of structure. Yet when we introduce the calcium, it will change the folding structure of comadoline, and that change of the folding structure will affect on the GFP fluorescent activity. So it will enhances its emission for light, and that can indicate the binding with the calcium. Yet this GCAM does have a few challenges. The first one, it's a big protein. It can be not easy to express it within the cells. And the second thing, because GFP is already a fluorescent material, then once calcium bind with it, intensity just enhances. That means there will be a noise to signal ratio. The background can be blurry, and the data could be affected for interpretation. Now, to move on from that, I looked up latest publication that was published in 2022 for Nature. They introduced the concept of photo-switchable activity for the GCAM. The reason why they wanted to do photo-switchable, they wanted to block or change the wavelength of GFP before and after binding with the calcium in order to prevent the signal to the noise ratio. As you can see here, when they introduces the calcium, the wavelength shifted from 400 to the 500 nanometer area. That can be a good thing, then they can filter out the image afterward. But still, there is a limitation for the size of this protein. It's inconsistent for the expression level. Also, the detection limit for this big protein to the calcium, it can be problematic and can fluctuate between one experiment to another. So to do that, I will talk about what I wanted to do or how I can design something new for a metalloprotein. We needed to start with the three main things or three main key points. First, the metal we wanted to target and the coordination number related to it. The second thing, as you can know, that the metal, each one of them does have a different size and different coordination number. And based on that, we can pick up what kind of a residue of amino acids that can bind with it or surround that metal. And then based on that, then the protein dynamics and the structure of folding that can be done surrounding that metal. And it can be used for the function itself. So these 3 points, I'll talk about it, and you can see it's the main thing I did throughout my experiment. Now, the fusion between, one thing, again, I wanted to introduce you to, fusion between organic molecule and metalloproteins. Now, metalloprotein itself, it does work, but we can go beyond nature. And to go beyond nature, we can introduce a small molecule or synthetic within the naturally occurred proteins that can enhances the activity of that protein. So as you can see from here, these three peptide bundle with the metal itself, once we introduce porphyrin in the middle of it with a certain metal, then the structure is more concise, and we can control that activity based on the organic molecule we choose. Now, metalloprotein hybridized with the protein and hybridized with the small molecule is already well done, but usually the activity could be separate between the small molecule and the peptide or protein itself. So what we wanted to do is combine all these three: metal binding, small molecule activity, and the protein function as well into our project. So this is, I'm going to talk in general about the design idea of my work, and then I'll guide you through a step-by-step for why we designed each one of them. We started from the comadoline, which is a well-known calcium binding indicator. I explained it in the GChem. Now, when we digged in deeper into the comadoline structure, we realized that it does have 4 calcium binding motifs. And on the upper part, you can see that these motifs are kind of intertwined with each other in position, means that they are opposite side or C2 symmetry from each other. Now, what we wanted to do is we picked up the calcium binding site from one of these motifs, And we looked it up, that truncated that one, and then conjugated with the myrocyanine, which is a photoswitchable moiety that I will talk about in the next slides. And then after computationally grafting both of them, the myrocyanine and the peptide, that has been done as a monomer form. Yet when we introduce the calcium, it can change its structure into a swap dimeric structure. The swap dimeric structure meaning that the one monomer is sitting next on the opposite side of the other monomer. Again, it's a C2 symmetry, mimicking a similar function for the GCAM calcium binding motifs. So basically, you can see that we do have two calciums, and the spiropyran with the peptides, they're opposite from each other. To design this part, we needed to consider three main things. The first one is calcium binding geometry and the pi-psi angle between the myrocyanine and the peptide, and the hydrophobic interaction between the two segments of these monomers. So I'll talk detailedly about each step, but this is the general story of the project. So calcium genetic indicator, the camadolin, as I explained, it does have 4 calcium binding motifs. Now these motifs, they intertwine from each other in a C2 symmetry. And because there are 4 calcium binding motifs, when we wanted to pick which segment of these short peptides we can use, we overlapped all these 4 of them, one over each other, and we realized that the calcium binding sites are similar in the backbone. So we just picked one of them, the first one, to proceed our experiment with. And then the small molecule or the organic moiety that we used into our project called spiropyran, it's a photoswitchable molecule. There are a lot of small moieties or organic moieties that can be photoswitched, but the spiropyran is a bit different in its activity because once we give it a UV light, the spiropyran will change its form into a myrocyanine, which is an open form, and that open form color will be different than the spiropyran is colorless. Color will change, and it does have this ionic oxygen within its structure. This ionic oxygen allow it to bind to a certain target, which is in this case the metal. Yet if we give a visible light to the myrocyanine, even if it binds to a metal or not, it will reverse back to the spiropyran. That kind of concept of this moiety we can utilize for our project in order to control the photo switching activity for our product, and that can enhance the imaging activity later on. So starting from the first step, as I mentioned, I did have 3 main categories. The first one is designing the calcium binding geometry. Each metal, as I mentioned, it does have a certain geometry. For the spiropyran, it does have pentagonal bipyramid shape, which is 6 or 7 binding sites. It can be 8, but you can see when we conjugated the myrocyanine with the peptide segment that we designed, this myrocyanine elongated structure, it kind of trajectoried backward from the calcium binding moiety or the peptide. That means we cannot utilize it for a fitted structure to bind with the calcium. So as a monomer, this moiety cannot work. So we introduce it to Rosetta, which is a design program for peptide. And when we utilize it, we realized that Rosetta introduced a dimeric structure with a quite good energy that can be utilized for our design. And that Rosetta suggested that this myrocyanine form, the ionic oxygen, could be involved within the calcium binding geometry. And basically, we reshaped the whole segment into make it a fitted structure for the calcium binding sites. So in this step, we realized that a swap dimeric structure is the fitted one for our design. Now, to prove that, we went to another third point, which is the hydrophobic interaction. The hydrophobic interaction between these two moieties. As you know, if we have two dimeric structure, there needs to be some kind of a strong interaction to hold these two monomers within each other. For us, because the endpoint between the peptide from here with the myrocyanine, it needs to be a strong interaction in order to keep that geometry for calcium, or calcium binding site could be more rigid. So we introduced certain kind of residues like threonine, valine, and phenylalanine. By the way, as you can see, this myrocyanine will interact with X, which is one of these residues, and the opposite side would be the same. Once we did the cosine theta, which means the angle that can be between the phenylalanine interaction with the myrocyanine, we realized that the phenylalanine showed a better cosine theta compared to the other candidates, as well as for the binding energy, phenylalanine showed a better results compared to the other one. From there, we can see that the hydrophobic interaction between the phenylalanine and the spiropyran moiety is more effective. Now, to prove that if there's a dimeric structure, we run MD simulation. Now for the MD, we run it in two modes. The first one is without the calcium. And without the calcium, you can see that the two monomers are projecting away from one another. Yet when we introduce the calcium, these two monomers stayed stable for around 500 nanoseconds that we already run. So from there, we can computationally confirm that the swap dimeric structure is can be well established and well stable under the phenylalanine condition, or the design already that was hypothesized. Furthermore, we also utilized the MD simulation in order to validate the design. This is the model structure for the calcium geometry that was already introduced from the camadolin. It's already in nature. Now, what we realize is that the equilibrium structure after we introduced myrocyanine, it hold its similar geometry compared to the first one. So there was no issue in the way of mimicking the calcium binding geometry with the nature from camadolin. And this is a representation of I already showed you the MD simulation videos. The distances between each monomer were compared of with and without calcium. And you can see that without calcium, the distance was quite big between the two monomers. Yet when we introduced calcium, this distance has been reduced and they got closer to each other. Then to validate our design, we started with our experimental part. And first thing we had to do is synthesize the spiropyran with the peptide segment as a one monomer. Now, this synthesis has been done before, but spiropyran with the peptide was not much introduced. So we were able to successfully synthesize it and yielded around 85% of it, and then to validate whether spiropyran and peptide can work in aqueous solutions like PBS and give a good photoactivity. As I talked earlier, spiropyran has no color, yet when we introduced UV light, in time, the intensity started enhancing at around 510 nanometer, and you can see the color already changed from colorless to a reddish color with the time giving it the UV. Now, since it is a photo switchable, means we need to reverse it from open form to its closed form. And we've done around 10 cycles. Basically, it started from spiropyran and giving it UV, change to MC form, and then going back to close. For these 10 cycles, we realized that the absorbance activity or photoactivity of the spiropyran stayed the same. That is a good thing for holding on and moving with our project. So we need to test the calcium selectivity because and binding activity in the absorbance and fluorescence. Now, since it is a metal, we want to do the application of this work within the body or biological system that contains a lot of metals. We need to test its selectivity to calcium compared to the other ones. So you can see the spiropyran didn't have any kind of absorbance. And then once we introduced it to calcium, you can see that the shift happens from around 500 nanometer to the 410 nanometer with the calcium, and the color surprisingly changed to a yellow color, distinguishing calcium compared to the other metal. Now, you might be thinking, why is the lanthanide or lanthanum similar to the calcium in the color? Basically, calcium and lanthanum, they do have a similar geometry, and the only thing they are different and a little bit, the size is a bit bigger, but they do have a similar activity. So any calcium indicator or sensor that is synthesized, when they test it out, they need to compare with the lanthanum. They compete with each other. They do have similar activity, meaning if it binds to lanthanum, it can bind with calcium. So it should give a similar activity. So that was a validation for the photoactivity and the color change that happens when we introduced calcium. Later on, we tested out the detection limits for the absorbance and the emission. And as you can see in the absorbance, when we introduced calcium, the myrocyanine 500 nanometer wavelength started to reduce, and then the 400 nanometer started to increase upon introducing calcium in different concentrations. And then for the emission as well, it was around 500 nanometer, which is validated for application later on. And that intensity also enhanced upon introducing or increasing the concentration of calcium to our product. Now, we needed to test out the photoswitchable ability after binding with calcium, because later on we wanted to kind of guide or control the activity of our indicator within the biological systems. So we started, after it binds with the calcium, we started giving it UV light, and it binds with the calcium. And then when we introduce visible, it reduces it. It goes back to the closed form, so there's no binding. So within the 5 cycles, in around a couple of seconds, the binding happened, and also the photoactivity stayed the same, meaning that it is good for us to keep using and control our product later on. Now, even though we've proven the photoactivity that it binds with the calcium, we need to prove it from a biophysics point of view. So I tested it out with the ITC, comparing this calcium with our indicator to the other metals that was tested. And you can see only with the calcium and lanthanum, it binded compared to the other metal that showed no binding. Basically, you can see the KD value is around 0.7 micromolar to calcium and 0.1 with the lanthanum. Basically, lanthanide, because of its size, it is supposed to have a higher binding affinity compared to the calcium, yet it also validates our selectivity that was done with the absorbance for these metals. Now, furthermore, now that we've proven all these selectivity and binding activity to calcium, now we need to validate our structure. Does it really make a dimer? Is it really swap dimer? So now we start to analyze that part of the project. Now, the first thing we wanted to do is test out whether it's a monomer or dimer. We wanted to check it with the size exclusion chromatography. Basically, when you run the size exclusion, you can see that the FPLC at 25 milliliter, this is the monomer size. And once we introduces the calcium, the size of this complex started to enhance and giving the dimer structure, and this size started to increase upon adding extra, equivalence of the calcium when we increase the concentration of it. That validates the size and doubled up for spiropyran and peptide for the calcium once it binds with the calcium. Later on, now, even though the size exclusion gave us a hint about our structure, yet to getting in deeper, we checked the NMR. And for NMR, in this case, we checked DOSI, which is a self diffusion coefficient NMR. Basically, we measured NMR at different equivalents or concentrations of calcium, and then we picked up the calcium binding sites. And then based on that, we calculate the diffusion factor of it. In this and kind of NMR, basically the density of the our solution or our compound started to increase once the size increases. So you can see that before adding calcium, the density was a bit lighter, and then it started to increases upon adding calcium. That validates our size exclusion data, meaning that the size of our product does enhance upon adding calcium. Now, the other thing that we needed to validate with the NMR is, even though we designed the calcium binding sites and geometry, we needed to validate that structural wise. So we measured HMBC NMR, and from there we checked the calcium binding residue, and we can see that these residues did have a chemical shift of before and after adding calcium. So since all of them shifted, we hypothesize that this is due to the calcium interaction and the binding that can change the angle or the interaction between these residues with the metal. Now, at the last step of our work, we wanted to validate our final structure. And we, as a biologist or chemical biology, we always aim to get the full structure of our compound, designed one, either by crystal or cryo-EM or NMR. Due to the size of my molecule, it's quite small. NMR was the ideal way to get the final structure of our compound. What we did is just a simply way I will run throughout this process, basically measuring NMR, and then calculate the inner protein distance by the panic method. And then those distances that was extracted from the NMR data was run through an MD simulation, and it gave us a few trajectories that we can run with. And after that, we can pick the ideal structure that can validate our design. So the first step from that process that I talked about was measuring ROSY NMR. And from measuring ROSY NMR, we were able to identify the positions that we wanted to calculate the distance from. And after that, we used the panic method. Basically, we exported the 1D ROSY from 2D ROSY. We exported to a 1D, and we calculated the NOE trajectories with it. And we were able to get the distances between the positions or hydrogens that we wanted to. And after that, we were able to implant it on this calculated distance within the MD simulation, and we got the trajectories. We ran around 30,000 structure trajectory. 5 of them were matching up with our structure, and the identifying was between 99% matching with our design structure. So with that being said, to summarize this whole process or the finalized structure, this is in the gray color, you can see is the design structure, and in the orange is the NMR-driven structure. Now, because of our work, we wanted to validate two main parts: is it swap dimeric? And second one is the coordination geometry really does match our design. From there, we can see that it did match, and the distances between the hydrophobic interaction of one monomer to the other one, it matched with our already designed one. So that was a good proof for our work. At the end, since we wanted to do the application, and we introduced a new functional molecule that can be a hybrid between a small molecule, which is a spiropyran, and a short peptide segment that can bind with a metal, yet it can give a different kind of a structure or unique structure that was a swap dimer. We wanted to first publish this work as it is for now, and currently we are in the process of preparing the application work for our second publication. So In the summary, we were able to develop a calcium and light switchable peptide hybrid, and the CAPS, which is calcium binding switchable product that we made, has high fidelity and affinity to bind with the calcium. And yet the swap dimer structure has been proven to be a hydrodynamic behavior, as well as this new nature mimic can be a groundbreaking for other complexes. And as I mentioned, currently we are in the process of the application, and the second paper is under preparation for now. So hopefully, when it's published, I can give you guys an update about that work. And at the end, I'd like to acknowledge and thank the professors who were with me, and mainly my supervisor, Young-Ho Kim. I worked with him for around 8 years. He was very supportive and open-minded to this work, and gave me the freedom to intertwine it. And he also opened the door for me to collaborate with two great guys, Bill DiGrado, who was a professor at UCSF. He's quite well known for his de novo design, and he supported the design idea and the shaping of this project, as well as Professor Kim Ki-hyun from pharmacy school at 성균관 University. I learned a lot from him, from the NMR structure. He was with me throughout that process, so I'm really thankful for them. And for my lab members as well, and my collaborators, Younghyun and Doohyun and Namhyun Kim, as well as for KIST and CNIR for funding this work. And at the end, thank you everybody for your kind attention, and I'm open for any questions you may have.